right. Welcome everyone to MIPIN's April webinar, Biological Control of Introduced Phragmites Australis in Ontario. We're pleased to be joined today by our presenter, Dr. Ian Jones with the University of Toronto. We'll give Dr. Jones a formal introduction in just a moment. But I always just like to introduce people to the Midwest Invasive Plant Network if you're not familiar with us. We are We've been around for about 20 years. Our mission is to reduce the impacts of invasive plants in the Midwest. I'd like to acknowledge our current sponsors. We are generously supported by the Invasive Species Center, Ecologic, Wild Type Native Plant Nursery, and Mark M. Holman, Inc. We're always happy to have new organizations sponsor us, so please be in touch if you'd like to do that. And I want to give a special shout out today to the Invasive Species Center of Ontario. They are a not-for-profit organization that mobilizes action against invasive species that harm the environment, the economy, and society. They are a hub for collaboration and knowledge sharing and a respected partner and leader in invasive species science, education, and action. And they have a great website full of a lot of great information on invasive species. So invasivespeciescenter.ca is where you can find them. So we appreciate them sponsoring today's webinar. And if you want to learn more about the Midwest Invasive Plant Network, you can visit mipin.org. There you will find a Midwest invasive plant list, information on invasive plant control, plant ID, tools for help you restore a site after an invasion. We've got directories for contractors and native plant and seed providers, a SISMA directory so you can find local folks who can help you with your invasive species questions and management. We can link you to the Woody Invasives of the Great Lakes Collaborative, which is another great resource. You can also find out about our annual conferences. Our next one is coming up October 6th through 8th in La Crosse, Wisconsin. That's the Upper Midwest Invasive Species Conference. So hope you can join us for that. And finally, we just hope you'll stay connected to us. We have a quarterly newsletter, email listserv, an events calendar, social media pages, or you can always just drop us an email with any questions that you might have. And then finally, we just appreciate you being with us today. We hope you'll consider supporting our outreach and education efforts. While we have sponsors, we also rely on individuals for support. So there's a QR code here. You can go to mipin.org and use our donate button. And I will also drop a link in the chat once we get underway. Okay, well, that is all from me. I'm going to go ahead and invite Dr. Jones to share his slides while I give him an introduction here. So again, pleased to be joined today by Dr. Ian Jones. He is a research associate working in the Smith Forest Health Laboratory at the University of Toronto. His research focuses on the ecology of insect-to-insect and insect-to-plant interactions. He received a master's degree in applied entomology from Imperial College London and completed his PhD at Florida International University. His current work focuses on classical biological control of invasive plants. Through an understanding of the ecology of biological control agents and their interactions with native species, he seeks to improve the release strategy and establishment of these beneficial organisms, contributing to a more effective and sustainable control of invasive weeds. So thank you again for joining us. Please go ahead and proceed whenever you're ready.
Yeah, thank you very much. I'm just actually going to stop my video to make sure that I don't get any kind of buffering during this, but let me know if any issues come up. Yeah, so thank you very much, everyone, for having me today. I'm delighted to be here to talk about classical biological control of invasive weeds in Ontario, but also some of this work moves over the border into the US. So I'm actually going to talk about two different weed biological control systems today. The first one being Phragmites. And that's the system that we're working kind of most intensively on at the moment. And in the context of Phragmites, I want to talk a little bit about what classical biological control is and what we can expect from it, and importantly, how we make it safe. Then I'm going to give you a kind of broad update on the Phragmites biological control program. And I'm going to talk in a little bit more detail about some methods we're developing now to actually transfer insects from existing populations of these biological control agents that we're calling nurse sites into new Phragmites infestations, because this is something that's going to give us the possibility of relying less on lab-reared insects and being able to conduct far more releases and inoculate far more Phragmites infestations each year. And then I'll give you a bit of a kind of an overview of how we plan to use those methods to expand the program over the coming years. I'm then going to switch gears a little bit and talk about invasive swallowworts, for which you have a biological control agent too in the US, Hypena opulenta. And in that context, I'll talk a little bit more about some of the factors and some of the kind of roadblocks that can hamper the success and the early establishment of biological control agents. And with that in mind, I'll talk about some cold tolerance work that we've done in collaboration with Western University on our biological control agent for swallowworts, Hypena opulenta. So what is classical biological control? Well, Phragmites grows as a native plant in Europe. And in Europe, it plays a lot more nicely with other plants and animals, in part because it's there in combination with various herbivores and natural enemies that knock it back in its competitive ability. But when Phragmites is introduced to North America, it's introduced on its own in the absence of those herbivores. And it's that, in part, why Phragmites grows with such a kind of intensive, hyper competitive and invasive growth model here. So what we're trying to do through classical biological control is reunite Phragmites with some of its natural enemies and herbivores and knock the Phragmites back enough that it grows much more like a native plant here in North America. And so that's an important point to raise with classical biological control, which is that the ultimate goal here is not the eradication of Phragmites. That's not a realistic goal. What we're looking to do is knock back the competitiveness and the kind of the health of the Phragmites enough that we can allow native plants and animals a foothold back into these invaded environments. But what have we learned over the years in the field of biological control? What has taken us from kind of ecological disasters of the past, like the introduction of cane toads to Australia in the 1930s, to 20th century success stories like the successful control of purple loosestrife and St. John's wort? Well, the main lesson really that we've learned is the importance of host specificity of biological control agents. Cane toads are voracious, generalist predators that will feed on virtually anything that moves, and they wouldn't be considered for a second as biological control agents in the modern world. So when we set about looking for potential biological control agents for Phragmites, and this was something that was done in the late 90s in Switzerland, we were looking only for highly host-specific insects that fed only on that target weed. And fairly quickly, two insects rose to the top of the pile in that regard, and these were two noctuid moths, stem-boring moths, called Archanara neurica and Lanisa geminopuncta. As I say, both of them are stem borers. They go through one summer generation, spending almost all of that time inside Phragmites stems. And then as adults, they lay eggs underneath the leaf sheaths of the Phragmites stems, and those eggs are the overwintering life stage. When we identified these insects in Switzerland, we strongly believed them to be highly host specific on Phragmites, but nevertheless, we put them through over a decade of rigorous host range testing. And this host range testing is what forms really the cornerstone of modern weed biological control. And this is the reason why in recent decades there have been no examples of non-target impacts of weed biological control agents. So how this works is we use something called the centrifugal phylogenetic method, whereby we develop a list of test plants, starting with the plants that are most closely related to the target weed, so Phragmites in this case. And then that list expands to include plants that might not be so closely related, but that might have some economic or cultural significance. With each of those plants, we then conduct experiments to show whether our candidate biological control agents can feed on those plants at all, whether they can complete any part of their life cycle on those plants, and whether they might be able to lay eggs on those plants. And it's only when we're completely satisfied that these candidate agents are highly host specific on the target weed that we will go ahead and petition to release these insects into an introduced range. And as I mentioned, this process for Arcanara neurica and Lanisa gemina puncta took 11 years. So it's a highly rigorous process. It started in Switzerland and then moved to labs in Rhode Island and Cornell. And only then did we actually petition to release these insects. What that host specificity testing looks like starts with fairly simplistic no choice experiments that you can see on the left that usually happen in small enclosures. And we're taking the candidate biological control insect and essentially saying, can you feed on this test plant or will you just die? And then later on, as the testing progresses, those tests become more complicated and realistic until ultimately we're conducting field trials, which much more closely mimic the kinds of environments that these insects would be released into. In 2019, we received permission to release both. And since that time, we've released about 40,000 of these insects across 60 sites in southern Ontario. And so this map gives you a pretty good overview of where those sites are and the spread that we have across the province. So some of those releases were 5 to 6 years ago now. And in those early release sites, starting to see exactly what we would want to see. This is a site at Rare Charitable Reserve in Cambridge, Ontario. And you can see a before and after picture in which the Phragmites now looks significantly shorter and less vigorous, less competitive in terms of whether we're seeing native plants recovering in these sites, but that's something that we're hoping to start generating over the next few years. But certainly we're very happy with where we are at this stage in the program. And the way this has been achieved is entirely through the release of lab-reared insects. So these biological control agents are reared in Switzerland at Cabby and also at the University of Toronto. As I mentioned, they have one summer generation. They then lay eggs, which are the overwintering life stage. So at this time of year, what we have in the lab is a load of Petri dishes full of biological control agent eggs. Those eggs are then released in one of two ways. They're either released as eggs, as you can see on the top row. So the eggs are placed inside this internal container. When they hatch, they then crawl out of the internal container and drop through the mesh at the bottom of the larger container, and then move on to the growing Phragmites from there. The second way we release these insects is to allow them to hatch in the lab, and we then use a paintbrush to inoculate individual caterpillars into cut Phragmites stems. And then those stems are then placed into these florist foam blocks in these sort of bouquets that then go out into the field. So egg releases are very easy to conduct. Because the eggs are the overwintering life stage, we can conduct them anytime during the winter or early spring, but they're much less effective. The larval releases are by far our most effective release type, but as you can see, they're far more labor intensive and they can actually only be conducted during a relatively short period in early spring when the eggs start to hatch in the lab. And so there are kind of limitations to both of these release types, and they also both require lab-reared populations of these insects, which is a kind of time-consuming and expensive process. I showed you that kind of dispersal of different release sites across the province. And among those 60 sites, we now have a fair few sites that we would term nurse sites. So these are sites that we've been going back to for 3, 4, 5 years and continuing to see larger and larger populations of these biological control agents. We expect to see a certain amount of natural dispersal of these agents across the landscape, and we're monitoring them so that we understand their dispersal behavior better. And that's kind of indicated here through these green arrows. But what we also want to do is help these animals along by manually moving them from nurse sites into new locations. And in so doing, massively accelerating the rate at which we can get more and more Phragmites infestations inoculated. So the big question here is, if we have a population of these insects in the field, what's the easiest and most effective way to actually transfer individuals from one site to another? They're not beetles that feed on the leaves that we can just kind of sweep net. We have to be a bit more creative in terms of how we actually move these insects from one site to another. Now, I talked a little bit about the life cycle of the insect, whereby they have one summer generation, then the adults lay their eggs underneath the leaf sheaths of the Phragmites stems, and they stay there over winter on what ultimately are standing dead Phragmites stems. What you're seeing on the left here is 100 Phragmites stems that were collected from the University of Waterloo, one of our nurse sites. We took those 100 stems back to the lab and carefully dissected them to see how many biological control agent eggs we would find on those stems. And we found 316 eggs on 100 stems. So that gave us a lot of confidence that this is a legitimate way to harvest these insects. We can find them fairly consistently and effectively by harvesting Phragmites stems. What this graph on the right-hand side shows is data depicting where on those stems the eggs were found. And the vast majority of the eggs were found in the lowest 1 meter of the Phragmites stems. So this is really important to us because Phragmites stems can be almost 5 meters tall. And transferring a 5 meter tall stem from one site to another is not an easy process. But what we know from this data is that we can collect just the lowest meter or meter and a half of these Phragmites stems, and in so doing, we'll be harvesting the vast majority of the eggs on those stems. So in the summer of 2024, for the first time, we piloted this stem transfer release process. So a number of stems were collected from 3 different nurse sites across the province. And then they were transferred into a recipient site at the university, at Ontario Tech University. So Ontario Tech University has these planters in the middle of the campus that for many years have been inundated with invasive Phragmites. So we mowed that Phragmites in the winter, and then we transferred in 18 blocks of 75 stems that had been transferred from nurse sites. Those transfers, though, were conducted in 3 different treatments based on when we collected them from the nurse sites, when those stems or where those stems were overwintered, and when those stems were transferred into the recipient site at Ontario Tech University. So in treatment 1, the stems were collected from nurse sites in the fall, shortly after the eggs would have been laid, and then they were transferred straight into Ontario Tech. So those stems and any eggs on those stems would have overwintered in the recipient site. In the second treatment, we collected the stems from nurse sites in the fall, but then we overwintered them in sheds outside the lab at the University of Toronto before transferring them into Ontario Tech in the spring, shortly before the growth season. And then the final treatment, everything was done in the spring. So we collected stems from nurse sites in where they'd overwintered and then transferred them directly into Ontario Tech just before the growth season. So what you're seeing here is what those planters at Ontario Tech look like in the summer when the Phragmites is all grown up. And we monitored the Phragmites surrounding those 18 different blocks of transferred stems. Now, larvae of these biological control agents don't travel more than about 1 meter or 2 meters. And so any damage surrounding those transferred blocks we know is associated with eggs that were in those particular transferred stems. And what we found was surrounding all 18 of those transferred stem blocks, we saw a significant amount of damage from these biological control agents. So this is really, really encouraging to show us that we can move these insects into new locations through this stem transfer technique. What we also found from this experiment was that there was no significant difference between blocks of stems that were collected from different nurse sites. So we're getting pretty consistent results regardless of where we collect the stems from. We also didn't see any significant difference between those 3 treatments that I described. And what that means is that not only does this method work, but we have a huge amount of flexibility potentially in terms of when we collect the stems, how they're stored over winter, and when they actually go into a recipient site. So this is great for us because it makes it easier for us to conduct these releases, but it also hopefully will make it easier for us to bring other stakeholders into this process. For example, if conservation authorities wanted to actively conduct these releases themselves using nurse sites that we had identified for them. So as we continue to expand the Phragmites biological control program, our method of doing so now it uses this stem transfer technique at its core. And so, for example, in the summer of 2025, just gone, all of our lab-reared insects were released over a relatively small area in Durham region. The idea is that we will monitor those sites, and as they become useful nurse sites, they'll then be used to redistribute insects throughout that Phragmites management area. So each summer, we're looking to identify a Phragmites management area and then furnish that area with nurse sites that can be used to transfer insects across the Phragmites management area. So for 2026, in a few months' time, we'll be starting on releases in Essex region. We have 8 or 9 sites identified there. And in the summers of 2027 and 2028, we'll be looking to identify Phragmites management areas in the far east and the north of the province. So we're looking to just kind of to develop these nurse sites in areas that will allow us to spread most efficiently these insects across the province. So a kind of nice announcement I can kind of make about Arcanara neurica and Lanisa geminopuncta is that after 5 years of releases and 5 years of consistently seeing successful reproduction and overwintering of these insects, we're now happy to say that by the most conservative measure, these insects are established in Ontario. And as I've mentioned, the transfer of stems from nurse sites into new locations is likely to be a big part of the process of us expanding this program over the next few years. So I want to switch gears a little bit now and talk about invasive swallowworts or Vincetoxicum rossicum and nigrum or dog strangling vine, as we call it in Canada. So, as many of you are, I'm sure, aware, we've had a biological control agent available to us for quite a few years now in Hypena opulenta, which is a foliage-feeding moth that then pupates in the soil and then emerges as an adult in the spring. These moths were released in Canada initially in 2013 and a bit later in the US in 2017. In both of those locations, we have successful establishment of the agents in as much as we're seeing damage from the agents year on year. Particularly in Canada, we've seen damage over a period of 7 or 8 years, and we've seen damage several miles away from the original release point. However, in all of these situations, we've never seen these insects reach a population high enough to do any real damage to the weed. They're not managing the weed in any way as things stand. And so we in Canada are no longer mass rearing and releasing these insects. As far as I'm aware, nobody's doing that in the US either. But what we are continuing to do is study the biology and the ecology of these insects to look at ways that we can potentially improve their impacts and to kind of generate some hope that in years to come we might start seeing some more successful control of these weeds. So when insects or biological control agents are introduced to a new location, there are several stumbling blocks that can prevent them from being immediately successful. And probably the two biggest ones are the activity of native predators and parasitoids and climate mismatches. And what I want to focus on today really is a potential climate mismatch between southern Ontario and the home range of Hypena opulenta, which is Donetsk, Ukraine. So here in Ontario, our winters are somewhat colder, only by a couple of degrees, and we do tend to get kind of higher snow cover. And so in conjunction with Western University, we've been conducting some lab work looking at the cold tolerance or cold hardiness of Hypena opulenta. So Hypena opulenta overwinters as a pupa underneath the soil. So it's the pupa that experiences the extreme cold temperatures of its introduced range. And so the setup for this experiment is that we have a pupa in contact with a thermocouple, all held in place with some cotton wool. And then that whole structure goes into the well of a cooling block. And then we watch as the temperature of that block slowly reduces. What we're looking out for is the supercooling point of this insect. That's the temperature at which ice crystals actually form internally in the insect. And we can see when that happens because the formation of those ice crystals creates a kind of thermal burst in the form of an exotherm. So the temperature at the base of this exotherm is the supercooling point of the insect. And this was measured on 35 pupae of Hypena opulenta. And we found a mean supercooling point of minus 9.13 degrees C. But the supercooling point on its own doesn't actually give us a huge amount of information about the cold tolerance or cold hardiness of an insect. It's only really informative in combination with knowing something about the cold tolerance strategy of the insect. So insects broadly fit into three different cold tolerance strategies. You have chill susceptible insects. These are insects that die as a result of just becoming cold. So they'll die at temperatures way above their supercooling point. So Drosophila is a good example of this. And generally speaking, they're not very cold hardy at all. The second cold tolerance strategy is freeze avoidant. These are insects that can't survive being frozen, but they can avoid freezing by drying out their bodies or accumulating antifreeze chemicals. So, for example, emerald ash borer um, is not freeze tolerant. It will die if it freezes, but it can reduce its supercooling point down to about minus 35 degrees C. So it can be incredibly cold without freezing. That final cold tolerance strategy is freeze tolerance. As the name suggests, these are insects that can be frozen solid for long periods and then thaw out and continue to develop in the spring. So goldenrod gallfly is a good example of that. So the way we characterize cold tolerance strategy in the lab is to run a very similar experiment to what I showed you before. But in this case, we put 16 insects into this cold block at the same time. So here you can see 16 thermocouples, each in contact with a particular pupa. We lower the temperature until 8 of those 16 pupae have frozen. So we're looking out for 8 exotherms. Once we've seen those, we whip everything out of the cold block and we bring it back to lab temperature, and then we monitor those insects for survival. Based on the survival of those insects, we can make an assumption about their cold tolerance strategy. So, for example, if all of those insects die, they're likely chill susceptible. All of the insects are dying from the cold exposure, irrespective of whether they froze. If all of the frozen insects die, but some of the unfrozen insects don't die, these are freeze avoidant insects. So they can survive those cold temperatures, but only if they avoid freezing. And then finally, if we see insects that freeze but then still survive, these are freeze tolerant insects. So this is the 16 insects that we were working with at Western University. We exposed them to this temperature regime until 8 of them froze. These are the 8 individuals in blue that froze and the supercooling points at which that happened. And then in this final column, these checks show the individuals that survived and successfully emerged as adults. So what you can see is a fair amount of overlap between the individuals that froze and the individuals that survived adulthood. So we can tell immediately from this that Hypena opulenta is a freeze-tolerant species. So we know based on these experiments that Hypena opulenta freezes at around somewhere between -7 to -10 degrees C. These are temperatures basically as cold as they're ever likely to experience underneath the soil in Ontario. But even if they were to freeze, we know that they can survive the freezing as well. So all of this evidence gives us a pretty clear sign that these insects can survive Canadian winters, and it's unlikely that cold is the factor that's actually limiting their success here. Some other experiments we've done looking at day length suggest that the relatively short day lengths in the summer here in Canada, and particularly further south in the US, compared to the longer day lengths in Donetsk, Ukraine, mean that these insects often go into early diapause here in Canada after only one summer generation, whereas in Donetsk, Ukraine, they regularly go through 2 summer generations. So this will be a reason why the insects' populations are growing very slowly, if at all. But all of this tells us that these insects, although their populations are growing very slowly, they can survive here, they continue to persist here. And there is hope that given enough time, they will reach populations at sizes large enough to have some controlling impact on the weeds. So I hope that's been kind of a fairly useful overview of 2 of the programs that we're working on at the moment. I mentioned that we're also working on Japanese knotweed as well. So if that's of interest, I'm more than happy to answer questions about any weed biological control systems that you're interested in. But thanks very much for listening, and I'm open to any questions.
Uh, Thank you so much. Make sure my mic's on. Yeah, so we do have some questions. I'll run through those, and if anyone hasn't submitted a question yet but would like to, just drop it in the chat for us. I'm just going to go back to the top here. There was a question kind of early in your presentation asking if you had any concerns about the toxicity of floral foam. I think that was from when they were being, the samples were
Yeah,
in the lab. Yeah.
yeah. Well, so that's an interesting point. So based on what we'd read, we didn't have any concerns. What we tend to do with those releases, we the insects are placed inside those cut Phragmites stems, and then they're placed in the foam blocks. That happens in early spring. And so then in the early summer, usually kind of late June, is when we'll come and do the first monitoring of any damage that's been done by the emerging biological control agents. And in that time, we'll collect in all of the cut Phragmites stems that we placed in those blocks. We'll take out because we're interested in how many we see exit holes in, because that tells us how many of those individuals that we released actually survived and moved on potentially to growing Phragmites stems. And then at the same time, we'll pick up those florist foam blocks and take them back to the lab with us. So they don't stay in the field for any particular length of time. Also, because we need those florist foam blocks to maintain moisture levels to keep those stems healthy enough for long enough. They're actually sealed within plastic containers. So they're in the florist foam itself doesn't really come into contact with the outside world a great deal. I do think if we put florist foam out just on its own, it might kind of disintegrate into the ground, and I would have concerns about that for sure.
All right. And then there was a question, what testing was done on native Phragmites?
Yes, so native Phragmites during the kind of the host range testing process was a big part of that process. And, the results of that generally are that the insects are able to feed on that native Phragmites. They can, in their early instars, feed on it. But because the stems of native Phragmites are so much thinner, it's very unlikely that a patch of native Phragmites could actually support a population of these insects. One of the other factors is that when these insects lay their eggs, as I mentioned, they lay their eggs underneath the leaf sheaths of the Phragmites stems, and it's under those leaf sheaths where they overwinter. In the native Phragmites, those leaf sheaths drop off the stem fairly early in the winter, and that would leave those biological control agents completely exposed where they would absolutely die. So I think there's a really nice kind of natural defense against overposition or successful colonization of these native Phragmites stands for these biological control agents. But it is true that it's possible that they can do some feeding on the native Phragmites. One thing we've started to do with our monitoring is that some sites where we've released these insects relatively close to native Phragmites stands, which are kind of increasingly difficult to find. But where we have done that, we will always monitor those native Phragmites stands to see if there's any damage from these biological control agents. And so far, there hasn't been, even in one case where the introduced Phragmites is literally growing into the native Phragmites patch. And we're seeing damage from the biological control agents on those invasive stems and none on the native stems. So it's a really kind of encouraging uh, distinction in their preferences between those two species. I think kind of certainly to me, the biggest threat to native Phragmites is the introduced Phragmites, and this is something that puts the balance of that competition back in their favor.
Oh, yeah, that's really interesting. So there's a question here. When might Phragmites biocontrol be available for public use? And kind of along those same lines, are there any plans to expand the program to privately owned properties?
Well, so for private use is a tricky one because it's hard. We think about this, and often that's the way that biological control programs go in the end. But this is it's a slightly tricky system in that it's not kind of a beetle where you could sell a box of however many beetles and kind of have them kind of sold commercially for people to release. The process of releasing remains kind of fairly complicated. It involves kind of lab rearing insects and then kind of inoculating them into stems and so forth. And so it's kind of difficult to imagine where it would be commercially, when it would become commercially available. I certainly wouldn't write that off. Um, but what I would say to anyone who's a kind of private landowner, many of our release sites are on private land because it's somewhat easier for us um, to kind of get permission to release on public land because anywhere we release, we're always looking for permission from any potential stakeholders. So public land is often the easiest way because we can be talking directly to the landowners. And so I will kind of provide my contact details. And if you're in a position that you're interested in the biological control, we can put you on a list for release as soon as we can.
Okay, that's really good to know. Okay, I think we answered that. Let's see. We do have a question here about Japanese knotweed biocontrol, but I think there's one other question here on Phragmites. So let's do that one first. Is it known why native predators of our native Phragmites have not transferred or had an impact on introduced Phragmites?
Yeah, so I mean, I don't know whether you could say it's known. There is a good study looking at kind of basically the whole suite of herbivores that feed on either or both native and introduced Phragmites. And kind of disappointingly, of those herbivores, some of them are native and some of them might be specialized on native Phragmites. We haven't seen a great deal of movement of those insects onto the introduced Phragmites. But many of those herbivores are themselves non-native, and the vast majority of those non-native herbivores are showing a preference for our native Phragmites over the introduced Phragmites. And so it's another kind of slap in the face really for the native Phragmites there. And so this is a kind of important way for us to redress that balance and have a herbivore that can really knock back the introduced Phragmites.
Yeah, that's a good question. I appreciate that. So do you want to talk just a little bit about which species have been tested for biological control of knotweed? Oh, did we lose you?
Yes.
Oh,
Sorry about that. So I only got the very start of that question, Michelle.
sure. Let me repeat myself. So there are a couple questions about knotweed as well. Do you want to spend a minute or two talking about which species are being tested for biological control for that plant?
Well, so that the knotweed biological control agents have been approved for release now, and have been for several decades actually. So there's a psyllid that was developed in the UK for release on various invasive knotweed species. So there are three invasive knotweeds really that are complex. So there's Japanese knotweed, Bohemian knotweed, and giant knotweed. All of the What we found is that this psyllid that went through lots of host range testing and was found to be highly specific to Japanese knotweed has now been released in the UK and in Canada and in the US. And in none of those places has it really persisted for any significant period of time. And we think it's probably high levels of predation on the nymphal stages of those psyllids. So they're relatively kind of vulnerable to all sorts of predatory mites and other small predators that we have here. And one of the issues is that there's quite a high level of specificity between strains of this psyllid and the different knotweed species. So, for example, there's a new strain that was collected from an area called Murakami that on the Bohemian knotweed, it caused the Bohemian knotweed to generate these kind of leaf curl galls. So where the margins of the knotweed leaf just kind of curls in on itself, and it creates a kind of a refugia for these psyllids to go into. And so in that situation, the psyllids are surviving much better, but they seem to need these kind of refugia that are created through the formation of these galls. And so we're still working on potentially finding the right combination of psyllids for the right knotweed species. And if we're able to do that, then I think we may get somewhere. But it's been a long process with not a lot of success, to be honest. The Phragmites biological control program is by a distance the most exciting one at this point, I would say.
Okay. So going back to Phragmites for a minute, has any testing been done specifically on hybrid frag?
No, so the hybrid frag is such a new thing, particularly in Canada. We're only now getting kind of confirmation that we think there are hybrid stands in Canada. So our goal now is to visit those hybrid stands for a couple of reasons. The first thing for us will be to do some lab testing to see whether our biological control agents will feed on the hybrid frag and whether they show any particular preference for it. But other things to do with the physics. Does the hybrid frag lose its leaf sheaths in the
Oh, you're cutting out a little bit.
no, come back?
Okay, I think you might want to repeat what you just said.
Okay. Yeah, so we're interested in bringing that hybrid frag back to the lab so we can do some feeding tests to see whether our insects will feed on the hybrid frag, whether they'll show any potentially for it. But we also want to know about the kind of physiology of that hybrid Phragmites itself. Does it retain its leaf sheaths over the winter? Because if it does, that means that our biological control agents can lay eggs on it and those eggs will survive. But at this point, it's a tricky one because we don't know how some hybrids that shed their leaves incredibly thin and support a population of the agents, but some hybrids that look more like the introduced Phragmites. And so that's something that we'll have to kind of continue to study over the coming years.
Okay. All right. So I thank you so much for repeating that. We do
No.
seem to be having some intermittent audio issues, so I think maybe will be best to sort of to wrap things up here. Um, this has been recorded, so people can go back and revisit it. Um, if people have follow-up questions, where would you recommend that they go?
Well, yeah, so you can, I will put my email in the chat. So you're welcome to contact me through that. What I would also say is a great resource is the Ducks Unlimited Canada website that has a huge section devoted to Phragmites and the Phragmites biological control team. And on that, you'll see a big kind of question and answer section that may answer some of your questions, but also you'll have contact details for myself and Dr. Rob Boucher, who works for Agriculture Canada and is another leader on the program. So yeah, those are all good avenues for getting hold of us.
Okay, that is a great resource to be familiar with. Um, Looks like we're getting some thumbs up, some appreciation for your presentation. I will echo that. Thank you so much for making the time to give this presentation today. We really appreciate it and you sharing your expertise. Um, yeah, so check out Ducks Unlimited. If you have any questions related to the webinar, you're also welcome to contact me at through mippen.org. And yeah, thank you all for spending your time with us today. And we have another Phragmites related webinar next month with Dr. Reed Smita from University of Missouri on the use of drones in managing Phragmites. So hope we'll see you again for that one. Um, Everyone, any final thoughts, Dr. Jones, before we sign off today?
No, no, well, thank you very much for having
All right. Thank you everyone for being here. Have a great rest of your day.
me. Bye.